Our next speaker for the afternoon session, Erin Bolstad. She's with the University of Montana and University of California, San Francisco in the field of cheminformatics. She's our presenter now on centralizing data for drug discovery in a multi-group academic setting. So I'd like to introduce at this time Erin Bolstad, please.
Hi. Okay, that works. My title is a little bit cumbersome, but really all I want to talk to you guys about today is how we've been using ChemAxon to solve a real big data management problem that we've had at the University of Montana, and talk about where we're at now and some of the exciting directions that we're going to go using ChemAxon. So primarily, I'll be dealing with data management and then how we're actually utilizing the data with the technology that we have. So a little bit of background first. The University of Montana, we have a biomedical and pharmaceutical department because we like long acronyms. And they're very interested in the central nervous system and particularly the glutamate cycle. And really all I want you to get out of this slide is that there are a lot of transporters and a lot of receptors, therefore a lot of targets going on in here. And we care about glutamate because it is the major excitatory neurotransmitter in the central nervous system. So dysfunction of this or irregularity of this has been implicated in a whole variety of either disease states or injury. So having some kind of modulation of the glutamate cycle makes for some really exciting therapeutics or research-based probes. The problem we have is that we have our little glutamate fiefdoms, which are just our research groups that are all kind of isolated amongst their own PIs and their own grant funding, all working on very similar targets, sometimes the same target, but for different reasons. And these guys face your standard academic issues in terms of where's my data, who's got the assay results, where's the spectra, so on and so forth. How are we going to organize this? And heaven forbid somebody graduate and not leave a zip disk behind them with something important. So we need a way to centralize this. And more importantly, we need a way to share the data across the little fiefdoms because everyone's working together. And if we can centralize this, the hope is that there would be new insights that could come from the mining of the data and new ways to look at it. Of course, you have your standard problems like proprietary data, who can see what's selective, who gets to input data, who doesn't. The system, is it easy to maintain because we really don't want to maintain a lot of stuff? Is it easy to use? So what we did there was the departments turned to the core lab for neuromolecular production, which then hired me to kind of solve this problem. So we went with ChemAxon because it was off the shelf, pretty easy to use. It's also highly adaptable. And so the rest of my talk is just going to be a story about how we solved this problem and how we used ChemAxon in order to do this. And it's kind of an ongoing story. We have a working infrastructure now that I'm keeping only to the CLNP until I'm sure that they can't break it before I deploy it to the rest of the department, which is hundreds of users doing all kinds of fancy pants things. So we started off with a MySQL based database, but that became pretty apparent quickly that we were going to need to switch over to Oracle and JChem cartridge, primarily because we're going to have multiple users, currently 12, but in the upwards of hundreds, accessing a single schema, this all sharing the same data or not sharing. And we also wanted the cartridge indexing features that come with JChem cartridge, particularly the chemical language via SQL. So right now, what I have deployed is IJC version 5.5 via Java Web Start. That lets me control the version, and everyone gets the same shared project, and they don't get to start their own little projects, or everything is really kind of rigorously controlled. So I'm talking just a little bit about the infrastructure and how we went about centralizing all this data. So all structures are put into a master structures table, which is got a cartridge index. We're also using this to catalog reagents, so it's kind of an inventory control as well. But a reagent is just simply flagged as a reagent so that you can filter on that pretty easily. Because it is a cartridge-based structure table, it's easy to look for duplicates and tautomers. And everything is, the parent key here is just a unique text-based ID that indicates if it is a U of M compound or a ChEMBL compound. Because when we put the data in here, we brought in all the ChEMBL data for the targets of interest for comparison and overall data mining. But we also had a lot of literature data that was not in ChEMBL, and then we had the stack of University of Montana data, be it spreadsheets, someone's lab notebook, or anything, just piles of data. So we had 2 poorly paid undergraduates who sat and put all this data into the structures table. And in future, this will either be put in by the people running the assays or the PIs, or the hope is to move on to an automated system once we get our plate readers set up and going. So after the structures, we have very specific tables, assay tables, 1 per target, a table for keeping mass spec data, NMR data. This is all entered via PHP web interface, a location and quantities table, and then a table with screening and docking results. Off base here is a users group table that we use to do a lot of lookups to see who belongs to who has access to what. Everything here is related to the structures table via foreign key and a many-to-one relationship. So basically, this is just saying you can't put any data in any of these tables unless the structure exists, and it's going to be a unique structure with a unique ID code. And a structure table is just your standard structures table with some physical data, but we also have the references, be it the literature source or the in-house synthetic scheme, the predominant target, what group, research group it belongs to, and when we get to row-level security, because we've got a whole bunch of security issues to deal with, there's a place to flag which group it belongs to or who can see it, as well as the reagent flag for further filtering. Our assay tables are just standard data tables that have every bit of assay data we could come up with that's vaguely related to it. But one thing that's come in really handy that I want to point out is that we have our assay values here, but if something was considered inactive, we put the upper limit of the assay and then a little Boolean flag that says greater than. So for calculations later, we can effectively just look at these little Boolean flags and say, well, that compound was inactive at that target for that assay. For those not familiar with SQL, I'm going to be talking a lot about triggers, and this is just a little stored procedure in the database that says when data is inserted, updated, or deleted on table X, fire this trigger and run SQL code Y. So triggers are an automatic processing unit. So the data flow that goes on here, that's been kind of our solution to pushing data around from the ChemAxon side of things. So Mr. User puts in data to the structures table. If it's not a reagent, this fires off a trigger that updates a summary table, and we'll talk about the summary table a little bit. Same thing goes for any of the assay data. As soon as anything is inserted, updated, or deleted, we do some math on the back end and update the summary table. Same thing for location and quantities. If this changes, the structures table is updated. That has the total amount, where it's located, who has it, can we share it, any column that you want to use for the location of quantities. And as soon as the structures table is updated, this fires the trigger again, and the summary table is updated. Surprise, surprise, when the screening and docking tables are updated, this also fires another trigger that all the data is pushed to the summary table. We have a web interface to update mass spec and NMR data, and of course, as those two tables are updated, trigger fires, updates the summary table. So it's a nifty little infrastructure, and I have the infrastructure is all set up, but what's in the little dashed boxes, these particular tables are not deployed quite yet because we're in the middle of server integration and getting new mass spec software. So we're just kind of keeping that back for right now until we get that established. But we have the infrastructure set up. But since I don't have the mass spec and the NMR stuff set up quite yet, just wanted to show what a sample of the NMR data and the mass spec data table would look like. Again, everything has a many-to-one relationship with a unique U of M ID or a ChEMBL ID if we're running compounds that have a ChEMBL ID. And the important thing here is that there's consistent naming and storage across the entire table. With the hyperlink function of IJC, we can link directly to where the data is stored on the server. So Mr. Experimentalist sits down, runs his mass spec or his NMR, then he goes to a web page and enters his Oracle username that generates a timestamp. He puts in the relevant required fields for experimental data and points to his file, which is then automatically uploaded to the CLNP server and named based on the group and user he belongs to. So everything's consistent. We always know where the Spectra data is. On the SQL side, we can insert the data in the hyperlink into the NMR and mass spec tables. And what's really handy is that if we flag it as clean data, then that passes to the summary table. So the summary table then has a link to the latest and greatest clean characteristic information there. And when I'm talking about all these triggers, they're not fancy. They're just compound triggers that run-of-the-mill kind of things that everyone's seen before, based on either passing data back and forth or performing some math functions. And what we get at the end of this was, as I talked about, the summary table contains all this pushed data that can be viewed in a number of fashions. And this is an example of looking at data. We can use a grid view that looks at only the U of M data, or we can look at literature and ChEMBL data, or combine it all together. And this is an example of what you would see through IJC in the summary table, looking at literature and ChEMBL only data. So we have our structures, which have been pushed from the structure table. And we have here, for CB1 and CB2, the averaged KI value. And what's reported in the et cetera column is the number of assays that were performed, the maximum, the minimum. If there was a standard error of measurement, then that's also reported there. And this is all calculated by the compound triggers. If one of these values was flagged as inactive, it passes the flag here. So it's very easy to look at it and say that this compound was inactive at both CB1 and CB2 based on these numbers and one assay. And since this is not calculated fields, it's actually real data in the SQL table, you can then further process it any way you please. We can look at log values for charting things. We can calculate specificity ratios. You can sort on it, which is the big thing. And then also perform your substructure searching, because it is not a materialized view. It is essentially a standalone table that is always up to date with the latest information. And this is another example. This is a U of M only data set up in the same fashion. And this is the way that I have it set up right now for people to use. But a PI could easily unhide particular columns, because they're going to be every column that we have pushed data for exists here. So we can, PIs can sit down and generate their own grid views or their own form views, which people like a lot. And we can look at the results for instance, the log CB1 versus CB2. We can organize the widgets any way we please. We can look at additional data, physical properties, stick in mass spec or NMR images. And since we have this is all trigger-based, we can just push the data into the summary tables, or the forthcoming ability to bind widget data from other tables, which I've heard is coming, which would be super cool. So security is the biggest concern here when dealing with this level of groups. Within each fiefdom, or within each little group, we have a number of roles that are played by people, PI, pharmacologists, synthetic guys, computational people, and a number of tables. Now, each one will have specific rights. A PI should be able to edit most things, or not at all. A pharmacologist can read structures, but shouldn't be able to edit them. Some tables we want to hide completely. Other tables we only want people to have insert and alter, but not delete properties, and so on. So in order to deal with this problem, I have authentication set up via the Oracle database and matching an IJC local user, which allows for some really tight control, and also allows for some nifty combos there. We can use IJC roles for hiding tables, who gets to edit the schema, who has administration access. On the Oracle side completely, we can then lock down privileges per user in terms of editability of tables, or generate entire SQL roles with table-specific permissions. And then on the combo side, we can use row-level security so that certain people or certain groups, or combinations of certain groups, can view certain compounds. And that's easy enough to adapt to sharing data amongst groups between roles and role level security, because sometimes groups need to share all their data together, but Group B may not want to share particular data with Group C. And this is actually a pretty easy system to integrate once you have the roles established and the user set up. So what they see when they sit down to look at their projects is, let's say Group A PI can see and edit the structures table itself. He can read all the other assays. We're assuming that everybody wants to share their assay data. He can write to his own assay tables, and he can write to the mass spec table, their NMR table. But let's say a pharmacologist from Group B, he doesn't need to be able to edit anything except his particular assay. So everything else is locked down, and some tables are even hidden from him. And that is how we have the data managed currently, how the infrastructure is set up. So the utilization of it is the really nifty part. And this is an ongoing story because there's a lot of stuff going on. So I want to kind of gloss over how we're going to be using ChemAxon to further the research we do. So for 2D virtual screening, we've got JCluster for training and screening set selections, integrated with ScreenMD or perhaps Mole Inspiration. We still like to try that one to determine parameter sets. And then once the screening is done, we can update the screening table on the SQL side. So how this would work is Mr. PI would sit down and insert either new data into the structures table, or he'd go in and flag screen at a particular target. Once this is flagged, it sets off a trigger that runs to the server and does the screening that we have set up, all based via either scripts or SQL commands themselves. Once we've got stuff calculated, that data is reported back to the screening table, which then fires the trigger into the summary table. It's the same, and what I have highlighted here is what we have currently, the bits and pieces. Most of it's there, we just need to kind of stick it together. The same thing goes for 3D virtual screening. I have a really neat, rigorous pharmacophore generation protocol set up that's all scripted, that has comes out with a three-dimensional pharmacophore of particular measurements, and we've used this approach to do some really neat drug design previously. And would like to integrate that with Screen 3D, not try and integrate it with ROX or Tripos Unity. Same kind of deal, determine the parameter sets for each target, a score cutoff for what we'd consider a hit or how potent. So PI sits down, inserts a structure, flags the screening box again, the trigger runs off, does his three-dimensional screening, either based on the pharmacophores or training sets that we determined elsewhere, update the screening table, fires the trigger, and the data is present in the summary table. We have all of these components together and set up, and the infrastructure is there. We just have to, again, plug it all together. Docking is another really useful way at looking at predicting potency and specificity. Because we're an academic unit, we have a lot of people who are doing experimental work on the target itself. There's no crystallographic data for a lot of these transporters. So we have homology models that are built with some experimentally observed constraints or some mutagenesis work in SAR. And we also have some really snazzy high-throughput automation scripts and data analysis set up. And all we need to do is script this into putting the results into a docking table. So same protocol, sit down, fires the same trigger. When you flag the doc column, it exports the data, docks it, brings it back in, reports the values to the docking table, which fires off the trigger for the summary table update. And what's snazzy about having the little HTTP in instant JChem tables is that we can then link to a JMOL of the docked solution. And again, we have most of the stuff all together. We just need to plug it up. So where is the future of all of this? We have Oracle working with ChemAxon, working with the data, with various security measures, and it all reports fabulously. It's kind of trigger-based. We'd like to bring in PubChem to complement our ChEMBL data. A lot of the virtual screening and the characterization and the docking are largely done. We just need to look at hooking them all together into the system itself. So the question then is, how has this addressed our overall goals in terms of data management, data utilization, and screening and docking and potency and specificity. Well, in terms of data management, the whole purpose of here was that the PI could come sit down and using any number of ChemAxon interfaces, instant JChem, JChem Web Services, or JChem for Excel, he can now do a lot of things he could never do before. Query, search, share data. He can view it in multiple ways that suits him best. He can make his own new grid forms. He could search subqueries, literature data, all at the same time, as well as the biological data. It's very, we have a lot of security measures now, so he can feel really safe that no one's going to steal his data. The familiarity of Excel is especially tantalizing for people who are scared to go onto a new platform. It's also kind of a self-modulating system, passing the data back and forth automatically, so no one has to sit there and say, oh, I haven't updated the assay data, give me a second. The whole point of this is that it is easy to view and understand, thanks to the off-the-shelf utility and the adaptability of particularly instant JChem. In terms of automated screening, what we're going for here is that the PI can sit down and he can do all kinds of snazzy things. He can input novel structures, we can enumerate leads, perhaps generate some new stuff using Reactor, or upload entire libraries like zinc subsets, or look at compounds for data sets that have no reported testing at a selected target. It might be functional. Stick this data in the structures table, flag it for screening or docking, and then off it runs to the server to do whatever it needs to do at whatever particular target. Server can do its calculations or rankings, relative comparisons to provide metrics that we can actually say this is predicted to be particularly potent or this is predicted to be particularly specific against targets X, Y, and Z. And the nice thing about this is because we're largely trigger-based, whenever there's new screening data that's updated, we can then recalculate the rankings so that the ranking you would see in the screening table is always up to date as compared to everything else that's already existing. And then the PI can sit down and see all of this. It's readily accessible right there on his computer. And tying this into the overall goal of understanding the potency and specificity amongst the academic group is now that we have ChemAxon and the data centralized to combine with the traditional SAR studies that are happening within the academic groups. Mutagenesis, we're doing some nifty fluorescent probe work, and the in-house pharmacophore-based binding site mapping going on. All these little fiefdoms can now share data as they see fit and lead to a better understanding of differentiating specificity and hopefully increasing potency of our leads or new compounds. And then once the system's up and working, it's easy enough to expand to other CNS targets of interest within the group. So thank you, NIH, for money. And thank you to my data entry minions who sit there and put the data in by hand painstakingly. And thank you, ChemAxon, of course, but I have to give mad props to my knights in shining armor, Tim Dudgeon and Peter Kovacs, who are the kings of IJC and cartridge, who helped me through a lot of the developmental issues that we were dealing with. The end.